Hi, everybody, and thanks for joining us today for our webinar, PGxome, the Power of Many. My name is Gina Landre. I'm one of our genetic counselors here, and I have the pleasure of introducing Dr. Jerry Machado, who will be presenting today. Dr. Machado is a laboratory director at PreventionGenetics in Marshfield, Wisconsin. He is certified in molecular genetics by the Canadian College of Medical Geneticists and also the American Board of Medical Genetics and Genomics. Dr. Machado earned his bachelor's and master's of science from York University in Toronto, Canada, and then his Ph.D. from the University of Toronto. He completed his postdoctoral fellowship training in molecular genetics at the Hospital for Sick Children in Toronto. Dr. Machado specializes in hereditary cancers and whole exome sequencing at PreventionGenetics. His talk today will be the PGxome, the Power of Many, and if you have any questions during the talk, please feel free to enter them and we'll address them at the end.
Great, thank you, Gina, for that introduction. So a little bit of background on our company. So PreventionGenetics was established in 2004. We are located in Marshfield, Wisconsin, which is in the upper Midwest of the United States of America. We've been in operation for approximately 13 years. We consider ourselves a comprehensive provider of germline genetic testing. We have a large test menu offering over 1,800 single gene tests. We have many panels, many next generation panels. We have deletion duplication testing for most of these genes. We do that by array CGH or MLPA. We also have a CMA array for constitutional findings. We consider ourselves to have unsurpassed quality in testing, interpretation, and reporting. We are a CAP and CLIA accredited molecular diagnostics laboratory. We pride ourselves in having exceptional customer service by having a team of highly experienced geneticists and genetic counselors, which we provide fast turnaround times and outstanding personalized services. So us and others started genetic testing by looking at single genes. As you can see from the diagram below, even though labs have remained relatively constant in operation, shown in red bars, you can see in the green bars, the number of disorders has grown exponentially since the early 90s. So since that time, we've moved from single gene testing onto next generation sequencing panels, which looks at 2 or more genes at the same time, thereby decreasing turnaround time and costs associated with genetic testing. From that, we are currently at the era of looking at whole exome sequencing, with the final thought of going on to whole genome sequencing. For exome sequencing, we focus on the coding regions of the genome. Most other regions we ignore. Exons make up about 1% of the genome, and this is important because approximately 85% of known disease-causing mutations are within the exons. So how do we specifically isolate the exome? What we do is we take genomic DNA using the SureSelect Technology by Agilent Technologies. We fragment this into smaller fragments, hybridize RNA baits, put this into a solution-based capture hybridization, and attach streptavidin magnetic beads. From this, we're able to isolate regions of interest, such as exons, and discard other fragments. From this, we wash the beads, we take this DNA, and then perform sequencing. Here is one of the approaches using Illumina technologies, where we take the genomic DNA, we attach adapters, ligate them, which then provides us with the sequencing library. From this, we put it on a flow cell, which we perform cluster amplification via bridge amplification cycles. From sequencing, what we do is we add fluorescently labeled nucleotides, one at a time, wash these as sequence is performed. Once we have the particular sequence, we take this and align it to a reference genome, which is used for varied interpretation. So there's 3 main objectives that I hope to cover today. So number 1 would be discuss the indications for our PGxome test, describe the exome variant interpretation process here at PreventionGenetics, explain the results of various PGxome tests. So we offer two main flavors of tests of PGxome. So the one I will be discussing today is the PGxome diagnostic test, but we also offer a PGxome health screen. And this particular test is intended for individuals who are basically healthy, but who want to learn their carrier status for recessive diseases or their susceptibility to adult onset disorders. So the whole thought of using an exome, such as the PGxome, is to end the diagnostic odyssey. So the PGxome diagnostic test is intended for healthcare providers who are looking for a genetic diagnosis when the clinical phenotype is unclear or previous test results have been uninformative. Specifically, this is ideal for unresolved genetic testing, such as when the patient has had normal karyotyping or microarray results, negative single gene or gene panel sequencing results, any disorders that have significant genetic heterogeneity, such as individuals who have broad phenotypes such as global developmental delay and intellectual disability, any individuals that have dysmorphic features, multiple congenital anomalies, or any other birth defects. The standard clinical practice often involves examinations for specific phenotypes, such as imaging, biochemical testing for inborn errors of metabolism, genomic tests such as karyotype, microarray, single gene tests, or next generation sequencing panels. But what's interesting to note is that once an individual has had these genetic tests for genetic diseases, approximately 50% of them do not have a specific diagnosis. So here's where whole exome sequencing comes into play, where we're looking at many different genes at the same time. So reported diagnostic rates from commercial and academic labs have found that whole exome sequencing assays have approximately 20 to 40% positive diagnostic rates. The rates are specifically higher for trio analysis, such as proband and parents, when compared to singleton analysis. And interestingly, what has been reported, there's a specific subset, approximately 5 to 7% of individuals who have whole exome sequencing tests have dual diagnosis, such as two non-overlapping clinical presentations. And it's important to remain current, as one recent study found that approximately 30% of genetic diagnoses have only been made due to recent literature reports, highlighting the fast pace of genes disease discovery and the need of genetic testing laboratories to remain current of medical literature. So things to consider when you're sending in an exome is the phenotype, to make sure that it's well defined, and also to note these things when sending in that information to the lab. Another thing to consider is the family structure and clinical status, such as we would like to know whether or not if the person is affected or unaffected for family members, whether sending in for a trio, quad, or if only sending in for a singleton, to make sure that the clinical phenotypes are clearly outlined. Another thing to consider is the secondary findings. So the American College of Medical Genetics and Genomics has 59 genes that they've outlined that may be important to test for whole exome sequencing. We offer this as an opt-out, and then we also offer another option, which is other Mendelian disorders, such as an opt-in option. How we do our variant analysis, we have two specific filters that we're using, so primary and a secondary. First, we want to make sure that the quality of our data is sufficient. So what we do is we look at any reads that are greater than 10. We also look at the genotype quality. We also look at the minor allele frequency, so anything that is 1% or above found in exact 1 kg ESV or other databases may be excluded from the analysis. We also look at the effect of the variant, so whether it produces a loss of function variant, such as nonsense frame shift or splicing variant. We look at missense variants. We may occasionally look at sense variants if they form a splicing defect. We also have a PG clinical gene list that we currently use in the lab, which is about 4,000 genes that we think that constitute Mendelian diseases and have been associated with some sort of phenotype. We look at PreventionGenetics past classifications of pathogenic, likely pathogenic, uncertain, and any missing interpretations that we do not have. These are performed by Ph.D. geneticists with disease-specific expertise. We also look at the inheritance pattern, so whether or not if the variant or disease of interest is inherited in a dominant manner, if it's a recessive, especially homozygous variants, if there's compound heterozygotes that have been found, and X-linked mode of inheritance. We also look at known rare pathogenic variants that have been reported in ClinVar as pathogenic or likely pathogenic. And lastly, we look at de novo variants. Our secondary filters, what we do is we exclude the minor allele frequencies, and we look at any past PG classifications of pathogenic, likely path, or uncertain for any variants that have been reported greater than 1%. We look at ClinVar of pathogenic, likely path. We look at variants that have the designation disease mutation in HTMD and our PG clinicome genes. We look at any loss of function variants in our clinicome genes. And lastly, we look at the ACMG 59 gene list for pathogenic and likely pathogenic variants. Our reports consist of 5 different sections. So the first one is variants in genes known to be associated with phenotype. Secondly, the variants in genes possibly associated with phenotype. Our third section is medically actionable variants, and this is normally included unless the patient opts out. These are the ACMG recommended list of 59 genes, which was currently updated in 2016 from 57. We also have our fourth section, which are variants in genes not associated with phenotype but may result in a Mendelian disorder if this is requested. So these particular genes have been put together, such as genes that are listed in OMIM, HCMD, and also other genes that we've curated from the literature or other databases. Lastly, we have a newly reported section of carrier status for variants that are causative for recessive disease, if that is requested. What is not reported in our reports, and often by other labs, are some genes and exons that are missing due to capture bias. Genes that are not medically significant or disease association is unclear. Paralogous regions such as pseudogenes and other homologous regions such as segmental duplications. Any disorders that are a result of specific repeats such as trinucleotide repeat disorders. Structural and copy number analysis, although we are currently working on this. Regulatory and deep intronic variants. And we currently do not report any mitochondrial DNA variants, but we do report mitochondrial disorders that have a nuclear genome component. What we request is that the proband sample be either DNA, blood, tissue, saliva, or amniocytes, only for prenatal fetal demise. Any additional samples such as parents or additional family members, a completed test requisition, signed informed consent, and especially important is the clinical notes and previous genetic tests, which can help us in interpretation. We specifically use the CRE version 1 probes, which is developed by Aryan Technologies. We look at the coding regions, plus and minus 10 base pairs. We use the Illumina's NextSeq 500 using 150 by 100 base pair pair-end reads. Greater than 97% of our target bases are covered at 10x or more. Our mean coverage of target bases is 120x or more. And all reported variants that you find in our reports have been confirmed by Sanger sequencing. What we found from our PGxome that healthcare providers have submitted, approximately 43% have been involved in neurological disorders, including seizures, other things including developmental delay, intellectual disability. Approximately a quarter have been multiple congenital abnormalities, 18% for neuromuscular disorders, and 14% of other disorders such as cardiac, immune, and skeletal disorders. From our diagnostic rate, what we found is approximately 40% of the samples that we have reported out have been positive, 40% have been indeterminate, and 20% have been negative. The indeterminate may include variants that may turn out to be positive, but currently at this time we were unsure and reported as such. And this is in line with what have been reported for other labs, especially for trio analysis. When looking at the specific mode of inheritance, we found that a little bit over half of our variants that have been reported in positive cases have been due to autosomal recessive disease, 41% due to autosomal dominant, and about 20% of those in the autosomal dominant have been due to de novo variants, and approximately 6% have been due to X-linked recessive disease. So when looking at secondary findings, approximately 77% of all cases have kept the opt-in feature for ACMG-related genes. We were not able to detect any significant secondary findings, so pathogenic or likely pathogenic variants in these genes to date. We have had 41% of individuals, healthcare providers, who are interested in having other non-ACMG related genes, but we did not have any healthcare provider that only wanted non-ACMG genes and no ACMG genes. So in total, we had 77%. What's interesting to note is that from our secondary findings, we found about 44% of our cases had some sort of gene that was, we found that was not in the ACMG list, such as those listed here on the screen, which cause CADASIL, which I will discuss in the short term, pseudoxanthoma elasticum, schwannomatosis, Brugada syndrome, and other cardiomyopathies. So some of these disorders may be treatable. So now I'd like to talk about some cases that we've seen here using our PGxome. The first case was a trio that was sent in for a 1-year-old male, and the clinical features included seizures, weakness of the limbs, profound developmental delay, difficulty swallowing, excessive sweating, abnormal muscle tone. A large epilepsy panel was negative that was performed by an external laboratory. It was also noted on the requisition that parents were consanguineous, but they were noted to be unaffected. So we performed the exome sequencing, and here is just an example of the alignment to the reference genome. And what we found was a particular variant in the DDC gene. And what we did was we specifically looked at an area of homozygosity, considering that the parents were related. This particular variant has been absent from the literature and population databases such as Exact. It is it's highly conserved amino acid, and some of the amino acid prediction programs that we use, such as CYP polyphen-2 mutation taster, predicted that this variant was damaging. We ultimately classified this variant as a variant of uncertain significance. DDC encodes an aromatic L amino acid decarboxylase, which decarboxylates L-DOPA to form dopamine and serotonin. Mutations in this particular gene cause this aromatic L amino acid decarboxylase deficiency, otherwise known as AADC. It's a very rare autosomal recessive disorder with approximately 100 cases been reported in the literature. And of note, homozygous missense variants have been reported. It is considered an inborn error of neurotransmitter biosynthesis. Some of the clinical features which were shared with our patient included hypotonia, dystonia, hyperhidrosis, otherwise known as excessive sweating, autonomic dysfunction, progressive encephalopathy with severe developmental delay. So mutations in DDT result in reduced enzyme activity of the AADC enzyme. And what this does is it leads to production of less dopamine and serotonin, as shown in the figure on the right. These are, the particular enzyme is involved in the production of several neurometabolites. And interesting, what we found was a little while after we reported this result, the lab went on to perform an AADC enzyme assay, and they showed that the proband levels were non-existent, thereby alluding to the fact that maybe this particular variant is likely pathogenic. Our second case was a singleton that was sent in. It was noted from a 1-month-old male who had polyhydramnios, hypotonia. It was also noted that the individual had breathing difficulties. They were especially concerned for congenital central hyperventilation syndrome. We performed the exome analysis on this individual, and we found two particular variants in two genes that were of interest. The first one was in the TTN gene, otherwise known as Titin. There's several disorders that have been reported for mutations in this gene, which relate to cardiomyopathy and muscular dystrophy and myopathies. They're reported for cardiomyopathies that in an autosomal dominant manner, where when related to myopathies, it's related to an autosomal recessive mode of inheritance. We found a splicing variant that was homozygous, and we classified it as likely pathogenic. Another variant that we found was in the BDNF gene, which has been associated with central hypoventilation syndrome. It's inherited in an autosomal dominant manner. We found a 2 base pair deletion, which results in a frame shift and premature protein termination. We classified this variant as an uncertain. I'll explain a little bit why these classifications were made for both these variants. So the TTN gene encodes titin, an essential component of sarcomeres. Sarcomeres are important for skeletal and cardiac muscle function. What they do is they provide structure, flexibility, and stability. They interact with other muscle proteins such as myosin and actin. Titin variants have been reported in autosomal dominant cardiomyopathy, but the variants that have been reported are especially dependent on the type of variant, such as a loss of function variant or a missense variant, and the domain location. Generally, they have been associated with missense variants, but other loss of function variants have been noted. As kind of shown here in this figure, the titin protein has several discs or bands, and there have been specific variants as shown as bars here within these particular discs or bands have been associated with cardiomyopathy, with most of the variants being clustered in the A band and not so much in the other bands or discs, as population cohorts or healthy volunteers are more often found in other bands outside of the A band. But the titin variants have also been reported less frequently in autosomal recessive myopathy disorders. Specifically, compound truncating and splicing variants were reported in central nuclear myopathy. This particular myopathy has displacement of the nucleus, which can be found using microscopy in muscle cells instead of the normally located nucleus at the edges of rod-shaped muscle cells. Some key features of this particular disorder include muscle weakness and atrophy, mild to severe breathing difficulties, hypotonia, but variable expressivity even within the same family has been noted. So some of these features, such as the breathing difficulties and hypotonia, were found in our patients. Generally, parents with children with central nuclear myopathy reported to be healthy and had negative family history for cardiomyopathy, but we're aware of one report where an individual had central nuclear myopathy who did have a cardiac phenotype. The BDNF gene, which encodes the brain-derived neurotrophic factor, we found a 2 base pair deletion, which causes a frame shift and protein termination. This specific variant has been reported in the population database of EXACT in up to 0.2% of individuals. However, only one missense variant was reported in the literature, and which was associated with congenital central hypoventilation syndrome. Interestingly, the missense variant who was inherited from the father did not have CCHS, but had hypotension and vasovagal syncope. So this begs the question whether or not if this particular gene or variant is associated with congenital central hypoventilation syndrome. Our third case is a trio that was sent in for a 3-year-old female. She was noted to have developmental delay, autism, intellectual disability, and speech delay. She had normal testing of Fragile X and other tests such as karyotyping and chromosomal microarray, and those turned out to be negative. We found one particular variant that was possibly associated with the phenotype in a gene known as RNF125, which causes Tenorial syndrome. It has been noted to be inherited in an autosomal dominant manner. However, the mother was found to harbor the variant. This particular variant was caused by a 1 base pair deletion, leading to a frame shift and premature protein termination. However, we interpreted this particular variant, even though it was undocumented, as likely pathogenic because even though no frameshift variants have been reported, missense variants that have been reported in one large deletion have been demonstrated to cause haploinsufficiency. So the clinical features of Aryan Syndrome, which is inherited in an autosomal dominant manner, include overgrowth, mild to moderate intellectual disability, which our patient had, macrocephaly, hypotonia, mild hydrocephalus, inflammatory symptoms. However, there are specific questions that still exist for the RNF125 variant. The mother was noted to be unaffected. There was no overgrowth mentioned for the proband or family history, but certain things to think about is whether or not if this particular variant has decreased penetrance, if there's variable expressivity in the family, whether or not this gene has, and variants that are pathogenic with this gene has an expansion of clinical spectrum, whether or not this is an allelic disorder. There's also the question of whether or not if this is truly an incidental finding. We also found in this particular young individual that they harbored a variant in the Notch3 gene, which causes CADA-cell. It was also found to be autosomal, it's known to be autosomal dominant. It was inherited from her father and from a well-reported missense variant that has been reported several times in the literature. We classified this variant as pathogenic. So CADA-cell type 1 caused by mutations in Notch3. CADA-cell stands for cerebral autosomal dominant arteriopathy with subcortical infarcts and leukoencephalopathy, so known as CADA-cell type 1. The specific key features are migraine, seizures, and stroke. It is typically found in individuals who are 40 years of age or older. So the father is currently at risk, and we also recommended an MRI, which may be able to show some of these findings before the stroke occurs. So there's definitely a genetic counseling component that is involved also with the exome sequencing finding results. So some of the conclusions that I have today are still there exist challenges in the interpretation of variants or genes that have not been reported. Individuals and their family members have secondary findings that expand on the FCCMG gene list. Whole exome sequencing can be used to end the diagnostic odyssey. It can be used to guide prognosis and improve clinical decision-making, such as targeted treatment, surveillance for later onset comorbidities, and palliative care. It can also be used for reducing the financial and psychological impact of diagnostic uncertainty. And lastly, genetic counseling related to recurrence risk and prenatal diagnostic options. So hopefully today I've covered the three main objectives that I discussed in the beginning. So the indications for PGxome testing, our interpretation process at PreventionGenetics, and some results that we've found here at PreventionGenetics. So our PGxome diagnostic pricing, for our individual pricing, we're looking at $2,490, a dual pricing of $3,490, trio pricing of $4,490. Any additional individuals that are added with no report would be $990, and any additional family member with a report would be $990. Thank you.
Thank you, Dr. Machado. So with that, we're going to wrap it up here, and we have a few minutes to answer a couple of questions that have come through. So if you have any questions that you haven't submitted, please feel free to send them through. Otherwise, we'll start with some of the ones we have. So one of the questions that came through, do we provide raw data?
Yes, so we do provide raw data in the form of BAM and VCF files. We will also supply variants of interest that we have found and interpretations for those variants to our clients upon request.
Okay, another one here. For the case that had the Titan variant, which band was that found in?
So that was found in the I band. So that, as you remember, the A band was the one that was related to cardiomyopathy. So the I band has been, the variants in that particular band have been reported in cardiomyopathy, but they have been reported also in the general population. However, some of the reports for central nuclear myopathy, they tended to cluster more so in the I-band, such as that with our patient.
Okay, it looks like we just have a couple more questions here. Another one that came through, does PreventionGenetics analyze genes of uncertain significance?
So what we do is we have our Ph.Ds and genetic counselors who scour the literature and databases, and we look at genes that we think can possibly have disease association, and we include those genes. Any genes that are outside of those, we currently do not assess, but at some point in the future, we may look at genes of uncertain significance.
Okay. Do you perform prenatal exome? Also, what is the turnaround time, and do you perform urgent exome on request?
Our current prenatal exome, what we do is anything with fetal demise. We don't do any ongoing pregnancies for using our PGxome. Our turnaround time, our maximum turnaround time is 45 days. We don't have a stat request, but we will prioritize exomes if noted.
And then it looks like we have just one other question here. Will PreventionGenetics report pharmacogenetic variants?
So currently we're not doing that, but we hope to in the next probably month or two to report that as an opt-in feature in the PGxome.
Great. Okay, I don't see any other questions that have come through. So with that, we'll go ahead and wrap up. Certainly, if any questions come about after this presentation, feel free to reach out to us. Dr. Machado has his emails there, as well as our general inquiries email, which is the best place to get a quick response on any of our any questions on our testing menu and specifically PGxome. Otherwise, thanks for joining us.